type of thing that we've done over and over again. Okay. Once again, if you know your enzyme names, for example, the dehydrogenase versus isomerase, that is half the battle, if not more. So first off, why do you need control for these metabolic pathways? Why? Why do you have to worry about this? Especially if you're going into medicine or doing anything biomedical, this is a very, very important concept to understand.
You could expend your resources too fast?
Exactly, that's one reason. What's another reason? You can cycle through resources way too fast. There are lots of possible answers here, people. What's another reason why you're going to want to metabolic control?
If you get an outside stimulus and it goes one way, but then you quickly turn it around, if there's no control, it'll just keep going one way when you need to reverse it.
Okay, so you can have futile cycles. Okay, what else? What are some of the biological, biomedical ramifications of things that go out of control?
Cell growth.
The uncontrolled cell growth is cancer, and that's actually a lot of these will spin up either an uncontrolled cell growth, stunted cell growth. You could go, delayed nerve impulses. There's lots and lots of possibilities, and this just shows you how so many of them are interrelated, and also the broad. As I mentioned before, there's only so many things. Yes, there's a 6-phosphate versus a 1-phosphate, or there's this little small nuance. But in reality, you're going to see the same type of thing over and over and over again. So once you learn the trends and you understand the logic behind it, trust me, that helps out so much. And it will also explain some of the logical questions later that are suggestive problems as well in medicine in general about, like, for example, why taking one type of drug may have a really weird interaction altogether, because you'll find out that there really are only so many ways to skin a cat, so to speak, is not good place to say. But these are the broad generic types of pathways that we have. We have carbohydrate metabolism, which what are some of the metabolic pathways that occur within carbohydrate metabolism? Glycolysis. What else? Gluconeogenesis. What's this? Citric acid cycle, and you could go into the pentose phosphate pathway and things like that. We have lipid metabolism. What's an example of lipid metabolism?
Fatty acid synthesis.
Fatty acid synthesis, fatty acid metabolism, or fatty acid metabolism. You can go into a lot of the steroids and all that kind of stuff there. There's amino acid metabolism that kind of states what it is. We actually won't cover the metabolism of cofactors and vitamins just because of the time. Like if we had like another class, part 3, we probably would go more into some of the secondary metabolites, especially those that have like really interesting diseases and orphan diseases and things like that. Then we have, what does it mean by biodegradation of xenobiotics? Up there at the top. What's a xenobiotic?
It's foreign. Some antibiotics you get from a foreign source.
Not even necessarily antibiotic, but yes, it just means it's a foreign something from without. And then we actually, I don't think we'll get into glycogen biosynthesis at all. And nucleotide metabolism, really, I doubt we have time to do that one as well. What about energy metabolism? What is the metabolic pathway of that? There's actually one, technically there are two, but they're coupled together. Oxidative phosphorylation, and electron transport chain, they kind of go hand in hand. Usually, there are times that they're not, they're uncoupled. Okay. So these are things that you should already know. If you do not know these terms, make sure you look them up and go over them. Things like homeostasis, flux, and cellular differentiation, which should be covered in A and B classes. Okay. So that's why I'm not, or Gen Chem. So that's why I'm not going to go into just definitions there, what flux means or homeostasis means. Okay, so then we're just going to talk about that all control mechanisms can fall under 4 broad categories. They don't necessarily have to be mutually exclusive, but they're going to fall within, I can't think of any exceptions, the 4 broad categories. All right. So there's allosteric, there's covalent modification, there's substrate cycling, and then there is genetic. And as I mentioned, they're not necessarily mutually exclusive. I think for the most part, it's pretty much self-explanatory. But we're going, I mean, just based off of their definitions, you can kind of get an idea, I should say. I shouldn't say self-explanatory. You can get an idea. So when we say allosteric, the very first one, what does that mean? What is allosteric?
Something binding to the enzyme, not on the active site, changing the conformation of the active site, which then like changes how the enzyme's reacting or if it reacts at all.
Okay. And so what's another word that we typically use for this, for allosteric? What? Cooperativity. And remember, it can be both positive and negative. I know we don't like, you know, whenever in other vernacular, if we talk about cooperativity, you don't even think it's positive cooperativity. But you could have negative allosteric, negative cooperativity as well. But it is, it's the binding of something is going to affect the catalysis, ultimately. Okay? So it can be both an inhibitor or an activator. What about a covalent modification? Covalent modification. Quentin, what does covalent mean?
It's a form of bonds.
Okay, so it forms a bond. So if you're covalently modifying something, what would be an example? We've actually seen this already.
If you add a functional group to a molecule.
Right, and what's one of the functional groups that we've seen added in the past? Not necessarily in the current chapter, or I should say chapter 14 specifically, but we saw it at the very beginning of the semester. What's one of the covalent modifications that you use a lot in these metabolic pathways? Phosphorylation, right? That's a big one, but it's not the only one. You can have acetylation, you can have methylation, you can have, or it can even be the dephosphorylation, deacetylation, demethylation. You can have an amination, that means an amino group is added, or deamination. You can have lots of different things. For example, with RAS, it has to be farnesylated, so the lipid tail has to be added onto it for it to be active under biological circumstances, at least. And so that's one that's there. So when I say acetylation, that's a very specific type of acylation. Okay? Acyl is just a generic. Acetyl means that there's only 2 carbons involved. Personally speaking, substrate cycling is the most difficult of the 4. Okay, especially, it's the most difficult to explain, I should say. So what do you suppose substrate cycling means for metabolic control?
Metabolite of that reaction comes back when it's in excess and inhibits the reaction from proceeding.
But that would be a feedback inhibition, which usually is allosteric.
Okay.
Right? Because that means you bind in case of the cycling. So, I mean, that is a form, but it's just not one for substrate cycling, per se. Like I said, this one is the most, for me at least, is the most difficult. I want you to read it and then think about it, and then we can. Glycolysis and gluconeogenesis is actually pretty. I think that's probably one of the reasons why they like to use this one as an example. By the way, it's also the one that I always forget. Whenever I'm trying to label, usually I forget substrate cycling. What would be an example in glycolysis and gluconeogenesis?
Hexokinase and
Hexokinase and this one I was going to use. What?
Glucose
6-phosphatase. 2 different enzymes utilize opposing reactions, and so because of that, they can affect the relative concentration of the substrate for the other. And many times they're going to be activated or inhibited separately. They can be compartmentalized differently. So like glucose 6-phosphatase. But that's the reason why, hexokinase can't work or is less likely to work well if glucose 6-phosphatase is overactive, right? Or is working full steam ahead, right? Because there's not going to be enough. Let me take that back. Glucose 6-phosphatase, if glucose 6-phosphatase is overactive, then it could possibly, depending upon the tissue, see that makes it even more complicated. Depends on the tissue, right? Because if we talk about the tissues that have glucose 6-phosphatase, then they're going to be futile. They're going to be competing with each other. If you talk about one that's not, so if we say, for example, what's one of the tissues that glucose 6-phosphatase is in? The lumen of the ER of the liver. Of the what? Liver, the hepatocyte. So for example, if we specifically talk about that cell's hexokinase, which we will later think this chapter, then it would be directly competing with each other. So, whereas if we're talking about hexokinase in the brain, an active glucose 6-phosphatase in the liver will actually help provide substrate for. Okay, and then genetic. What would be an example? A genetic control mechanism. Sometimes, one thought people forget about this one, and this is probably on the most simplistic, basic level.
The expression of certain genes sends down the pathways.
Well, or you can even say, you're right, the expression of certain genes, if you don't express the enzyme, it won't be working. And so literally, the amount of the enzyme present is going to depend upon its synthesis, right? So if you downregulate the expression of that gene, or if you upregulate the expression of the gene, you can actually affect the metabolic pathway as well. So typically, a lot of times this may be a little bit slower acting, right? Because it may be a little bit further downfield, versus covalent modification or allosteric, which is happening on target right there. So it's cycling is a little gray. It depends. Okay. Actually, we've been talking about this all of the last chapter, and so that's why it shouldn't be surprising. Glycolysis and gluconeogenesis, are they technically futile cycles, since they do compete against each other? So wouldn't it just be unproductive, or just lose energy? The physicist would say, oh, it loses energy due to heat dissipation, right? I was saying it wrong. Because any time that this is happening, heat's given off. Okay? I don't ever think, very rarely think in terms of physics, but we're just saying that wouldn't it be unproductive the moment you make the pyruvate, turns around and gets made back into glucose, that gluconeogenesis. So why are these not considered futile cycles? There could be more than one possible correct answer here, by the way. But why are these not considered futile cycles? Yeah.
Do we use the heat from that as...
We wouldn't typically hear, like for the ones that need thermogenesis, like babies or hibernating animals, the way that they actually, their heat's a little, and we actually will cover that later, is usually, this is not going to be the main way. Okay? So also, how is this not a fetal cycle?
Is each metabolic pathway regulated by certain hormones or something?
You could say, yeah, we're going to talk about hormones that do regulation. They're regulated by different hormones. They're regulated by different covalent modifications or allosteric modifications. They're regulated by different substrates. They can have different compartmentalization, which is the example we gave. Only certain tissues can fully undergo all of gluconeogenesis because of the fact that they are the only ones that have glucose 6-phosphatase, things like that. Kind of been pointing those out as we go along the line. First up, we're going to talk about hexokinase. This is actually one of the very important reasons why you must specify from here on out, from this point on out. So today, if one of the correct answers was hexokinase, and you didn't specify what kind, I wouldn't take it off. But from here on out, many times you may get at least partial credit deducted because there are different isozymes of the hexokinase. You cannot say glucokinase because glucokinase is specific for the liver and pancreas, by the way. I don't know why I need it. I don't put it on there. The liver and the pancreas. I think the same thing happens in your book. I think they primarily talk about the liver right here. Technically, by the way, just a little, it's kind of getting away to the end of the story. One of the chapters, pancreas also has hexokinase 4. And they do commonly call it glucokinase. It's very, very important that you know the difference between 1 through 3, which happens in the skeletal muscles and so on and so forth, and hexokinase 4 or glucokinase, whichever you prefer to call it. Okay? There are two very, 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 very important things that make it different with glucokinase. One is it has a much lower affinity for glucose than the other forms. So if it has a much lower affinity, will glucokinase have a higher or lower Km than the monocytic ones? Will the liver and pancreas have a lower or higher Km since it has a lower affinity?
Lower.
A lower Km? Does everybody agree or disagree? Anybody disagree? It's higher. Why? Why is Km, if it's a lower affinity, it's going to have a high Km? Think back to last semester.
You need more glucose to get it to the
Right.
maximum.
Okay. So therefore, remember, those are antagonistic. Not antagonistic. What's the word I'm looking for? Reciprocals. Reciprocals. So the Km for glucose on hexokinase 4 or glucokinase is higher than it is for the other forms. Hexokinase 1, 2, 3. And then there's a second one. Okay. What primarily happens in the liver, for the most part, yes, technically there's some that could happen to a much smaller scale in a couple of other tissues, but primarily in the liver, the other tissues don't do.
Storage of sugar.
It does store sugar, but to get that stored sugar for storage, what metabolic pathway does it do? Gluconeogenesis. Okay. So that being said, its form of hexokinase, it's sort of, I don't want to say self-sacrificing, but it will not use up glucose for its own glycolysis until after everything else, all the other tissues are taken care of. It's like the mama. No, I don't know. But whatever, it helps you remember. So because of that, its hexokinase 4/glucokinase, it actually is not inhibited by its product. All 1 through 3 is inhibited by its product. On your own, because time is ending, and I almost always ask this on a quiz or exam, or it's, and this one's just problems, is why does that make sense? That, and like what's happening on in the liver versus other tissues, one more way to explain that, that you want to have a higher Km, and the part that people many times forget is that it's not inhibited by its own product in the liver, okay, the pancreas. Okay, so, but, and then I know this is an old, I know they are transcriptionally regulated, genetically regulated as well. Okay, are there any questions?